All right, so let's introduce Anthea, who is here from Columbia University, and will be speaking to us on topological summaries of tumor MRIs she's shown improved survival prediction for glioblastoma multiforme.
Great. So very much for the invitation, and thanks to everybody for coming and listening to this. So what I'm going to present today, so first of all, this is joint work together with Lauren Crawford and Cheyenne Mukherjee from Duke, as well as Andrew Chen and Raul Rabadan here at Columbia. This work that I'm going to present today, it's not so much, I'd say that it's not new or sort of deep in terms of topological theory, so in math theory. There's statistics, but the whole idea of this talk today is to sort of show that this that we do in applied topology can be applied to other settings and come up with and I cover some really interesting, relevant, and pretty deep results. So that's sort of what I'm talking about today. So first of all, I'm just going to sort of present the motivating problem here. So the motivating problem is glioblastoma multiforme. It's a type of cancer. It's a brain cancer, actually. So it comes from a class of tumors known as gliomas that affect the central nervous system. And this brain cancer, GBM, is the most common and most aggressive in humans. So in 2016, there were about 15,000 cases, and extremely aggressive because the post-diagnosis survival is around 12 to 15 months. The probability of 5-year survival is less than 10%. And as far as treatment goes for for GBM, what happens is that you do surgery first to try and remove as much of the tumor as you can, which is difficult though because it's the brain. So first of all, there's surgery, and then you apply radiotherapy. So yeah, radiation basically, and then chemotherapy as well. And this has really good results. So basically, it increases your 2-year survival chances up to 26.5%. The thing to note about this therapy though is that this standard care is that there's no effective treatment for recurrent tumors. So basically, this is saying that there's really sort of only one treatment, and if you have relapse, then you can apply the same treatment to it, but it's not going to be effective. So to study this cancer, to study GBM, the data that are available are molecular data. So for example, this is mutation status, DNA methylation, or gene expression, and these are obtained by surgery. So when you take surgery, when you operate, you take a sort of a sample and then molecular assay, and then you can sequence and get all of this information. And other information that you get is imaging information. So these are, for example, MRI images that are obtained from radiology. So radiogenomics is a field which is sort of, well, it's radiogenomics, also known as imaging genomics, that aims to understand the relationship between clinical imaging data and functional genomic variation. So what it does is it takes images and data, and it uses the information from that to sort of inform prognosis, give you information to make clinical decisions, and basically things like that to try and, in the context of cancer, to try and treatment. So our existing results, it's a relatively new field, but there are existing results in radiogenomics. So in particular, it's been shown that including geometric information improves the predictions of survival outcomes. Geometric features can be used to predict GBM subtypes. So within GBM as a cancer, there's also subtypes of that. So the geometric features that are obtained from images can be used to predict those. They've also developed automated pipelines for GBM image analysis to link tumor geometry to patient prognostics. And then it's also been found that subtypes predicted by geometric features correlate with survival and the activation of molecular signaling pathways. Especially in the case of GBM, this idea of radiogenomics and trying to extract as much information as you can from images is something very valuable, because In the case of GBM, if you want to get molecular data, the only way that you can get that is by surgery, right? And so since it's a brain tumor, it really means craniotomy, which means that you have to open up somebody's, brain, like split their skull open, get the samples, and then stitch them back up. So that is extremely invasive. Imaging contrast is just really taken from MRI scans, so you don't have to go into anybody's skull to do so. The idea is really trying to extract as much information you can from the images to sort of inform the status of your disease. So I was interested in looking at these topological features. So the sort of looking at imaging information and extracting topological information and using this in radiogenomics. So when we first started studying GBM, it turns out that there are important characteristics of GBM which have a big role on the prognosis of the patient. So one is multifocality. So it means that you have tumor has multiple lesions. So your tumor arises from the same oncogenic effect, but it results in multiple lesions. So in the language of topology, which we're all familiar with here, this basically means connected components. So this is our H0 homology. Another characteristic of GBM is that there's that sometimes they exhibit necrosis as well. So necrosis is the existence of necrotic regions with a tumor, which are essentially dead cells in the tumor. So in in topological speak, this is really H2, right? You're looking at voids on the inside of this tumor. So the idea that I was really interested in was trying to find a way to integrate this topological information you can get from the images into radiogenomic analysis and see what that gives us. So when we got into this application, the final results of analysis was that we introduced a new statistic which summarizes the topological features of these tumor images in vector form, and in such a way that it's statistically targeted, so in a sort of robust way. And we actually did a full-scale, rigorous statistical analysis for imaging data against gene expression data. So with the aim to predict the clinical outcome, which is survival of GBM patients. Okay, so let's first talk about imaging representation. So what's imaging data and how is it represented? So in the past, images were represented as landmarks. So here on the right here, this is an example here of landmark imaging. So are the landmarks themselves, and they're superimposed on pictures of molars here. So we won't, in terms of what the data that we would get for shape statistics in the past, it wouldn't be actually the molars that you would get to see, but just these points. So landmark-based approaches, so basically to actually get these landmarks, they're user-specified approaches, so the users themselves pick out the landmarks that are important. There are also automated approaches, which basically tries to automate the specification of these landmarks. But landmark-based approaches, they suffer from structural errors when comparing highly dissimilar shapes. So the advancement of technology, we don't need to represent images anymore as landmarks, but we actually get them as meshes. So here is an example of an image representation as a mesh here. So this here is the representation of a brain with ventricles and as well as a tumor. This new representation of images that we have, there was a first topological approach to studying these due to Kate Turner, Cheyenne Mukherjee, who's co-author on this paper, as well as Doug Boyer from Duke. And they introduced this persistent homology transform. Before I talk about what the persistent homology transform is, I'm going to talk about what persistent homology is. So in classical topology, and in very sort of vague, basic notions, homology measures shape. So it does this by counting the occurrences of patterns. So in 3D, these are the occurrence of connected components, of loops, and of voids. And the idea of persistent homology is to adapt homology to point clouds. So point clouds are where we get data points that come from some finite metric space. The point of persistent homology is that we assign a parameterized sequence of social complexes to our point cloud, and we track how the homology groups change as the parameter increases. The output of this is a barcode, which is a collection of intervals. So you can also represent this as alternatively as a persistence diagram by taking these intervals as ordered pairs and plotting them. So essentially, barcodes can be seen as summary statistics of the data that we get from a point cloud. Just to sort of give you a visual representation of what these things look like. So barcodes, this here is an example from a simulation of a barcode where you see the black bars correspond to the H0 homology, the blue bars correspond to H1, and the green bars correspond to H2. And then if we're pulling each of the homology groups in terms of these barcodes as persistence diagrams for each of the homology groups, then we get something that looks like this. So this is just sort of to give you an idea what barcodes and persistence diagrams are. So the basic idea of the persistent homology transform, which was introduced by Kate, Cheyenne, and Doug, was to take these mesh representations that we have images now, and they're simplicial complexes. So the simplicial complexes, we can use the persistence diagrams as summary statistics of shape. So we collect all persistence diagrams of all dimensions for all orientations of the shape, and this is going to give you a summary statistic. Basically, mathematically, what this turns out to be is for a given simplicial complex, so this is your K, that's going to be your mesh representation of the shape, and then you rotate this shape over all directions on the surface of the sphere and collect the persistence diagrams that you get from that. Then this is going to be your persistent homology transform, which captures all of the information of your shape. So theoretically speaking, if you look at this definition here, what you're going to get is going to get infinite collections of persistence diagrams. But so because infinitely many directions on the surface of the sphere. So also prove stability, approximation error results, which tell you that you can finitely many directions. The error bound on taking finitely dimensions versus the infinitely many directions on the surface of the sphere, that's small. So for computational purposes, it's sufficient to many directions. So
in action, they're doing sublevel subpersistence. Is that right?
Yes, that's right.
Okay. Thanks.
Yes. So properties of this transform is that it's injective. There's a metric which can define distances between shapes, and because you basically just take the distance. So, for example, the Wasserstein distance, because there's a distance defined on the space of persistence diagrams, and it's a multi-scale topological summary. So, multi-scale meaning that you have a collection of persistence diagrams for each of the homology groups for each direction. So it's multi-scale in that sense. So this is a really nice way that you can sort of summarize your data, but it's not very useful if you want to do statistical analysis with this, because statistics can't really work with these multi-scale topological summaries. So I also developed the idea by looking at Euler characteristics of that, and we sort of took that idea and then we actually developed it further for a specific case of statistical regression models. So so we call it the smooth Euler characteristic transform, and it's based on the Euler characteristic. So the Euler characteristic is another topological invariant, and for simplicial complexes. So for example, if you take a simplicial complex in 3D, what it does is it counts the number of vertices, edges, and faces in an alternating sum. So your number of vertices, you subtract off the number of edges, and you add back the number of faces. So this is the way that you calculate Euler characteristic from mesh. And then if you're going to look at the Euler characteristic curve, so the EC curve calculates how the Euler characteristic changes with sublevel set filtration. So we took this idea and then we sort of mean-centered it, and then we integrated it. So this is sort of smoothing it, because when you're looking at statistical data, and especially large-scale statistical data is in the case of, gene expression data. When things get noisy, you want to have some sort of robust effects. And so these sort of the mean centering and then smoothing, these are sort of known methods in statistics that we apply to make your statistic more robust. So like the persistent homology transform, this statistic is also injective. You can also define distances on it. And as opposed to the persistent homology transform, what we have a vector valued topological summary. Okay.
So, Anthea, can you explain this in a bit more detail? So, what is A sub nu and B sub nu?
Okay, yeah, sorry. So A sub nu and B sub nu is the interval that you're doing your filtration over. Right. Your bottom line, and it's the top line basically.
Filtration values A sub nu and B sub nu. Right.
So the interval that you're basically doing your sweep over for your filtration.
So negative infinity should be A sub nu, is that right?
It should be A sub nu. So basically what we do is really anything outside of A sub nu and B sub nu is actually 0. So it really should be A sub nu. So I guess we were just looking at this really sort of, in terms of statistics, it was sort of more statistically intuitive to look at this as some sort of cumulant here. So you would accumulate normally from minus infinity, but anything outside of A sub nu and B sub nu is 0 anyway. So it really should be A sub nu and B sub nu. Yeah.
So the mean centered, or like to just get 0, or why don't we get 0?
What do you mean?
The color.
So this here is going to be the mean at every point that you subtract off of the value. So you take the mean. So you calculate everything throughout the whole filtration, right? And then you subtract off the mean value at each point. Maybe we can come back to this, but maybe it'll be clearer once I sort of tell you how we actually calculated this, like with a concrete example.
Okay, we're taking the mean.
What we do, so I'm going to explain this later, right? But what we do actually is we take the Euler characteristic value at finitely many sublevel sets, right? And after that, we calculate the mean of all these. And then we take that mean value, and then we subtract it off at every point. At every point that we calculate. So that.
So there were different samples, is that right?
Yes, it is.
Okay, sorry, I thought it was the mean over different values of X.
Oh, no. It's the mean over different samples. I'm sorry.
Okay.
Yeah, sorry. Okay.
All right, okay, and then the norm between these functions is just the L2 norm?
Yes.
Right, so the function on the sphere. And now nu here is a sphere. All right, thanks a lot.
Okay.
I think I'm starting to understand it.
Okay. Yeah, maybe it'll be easier once I sort of like explain how we actually concretely calculated with these tumor images. But I'm going to do that now. So.
Okay, great, thanks.
Okay. So first of all, what are the imaging data that we get? Okay, so we have MRI data, as I mentioned before, and what we want to do is segment them. Okay, here's an example. On the left-hand side here, this picture here, this is a picture of one of the slices that we get from an MRI image, and this red here is what the tumor actually looks like. To actually get something that we can apply the code to, which is something here, right, we have to segment. Okay, so we developed a segmentation algorithm which converts these MRI images here to grayscale, and we threshold in order to get binary images, and after that, we just segment morphologically to delineate. So it's sort of like a combination of automated as well as user-based segmentation here, because the automated was thresholding to create the binary images, but then it would be up to sort of like a radiologist or somebody who really knows what they're looking for to be able to segment and delineate the actual tumor. So here's an example here of one of the GBM tumors that you would see here. And what I was mentioning before, necrosis in GBM tumors. So you can see that here because you see all these black regions on the inside of this tumor. So this here, this patient here has his GBM, his tumor has necrosis here. So these sort of like the images that we get. So to actually build this this vector that we're going to integrate into our statistical analysis, what we have is we have these MRI images which are taken slices. So patient, we have about 25 slices of 2D grayscale images. So the number depends on from patient to patient because it depends on how big the tumor is and so. But we have about 25 slices of these 2D images, which we then segment using our algorithm. And then for direction, we compute not the transform, but we compute the smooth Euler characteristic at 100 equal sub-level sets of the filtration. This gives a vector of length 100. So basically what we do for this slice, for this one direction, is that we take equal sub-level sets, we get the Euler characteristics, we take mean of this, we subtract off the value at each time, and then we smooth. And repeat this 25 times. So we have once each of the 25 slices of the tumor. And what we do is then we average each component of the 100 vector slice-wise. So we do this over the slices. And output then is a vector of length 100, which is a topological summary for the tumor in that fixed direction. So if this is over 2 times over evenly sampled directions from 0 to 2 pi for each slice, then what we get is 72 vectors of length 100. We stack all of this together, and all output that we get is a 7200 vector, and this process is rotating the brain and then calculating topological summaries at each direction, and then collect all the information together. This is we did this for each of the tumors. So each patient then who has GBM gets a 7200 vector, and is going to be the summary of our topological information.
This is interesting. Can I go through some of it again? So the slices is D, and we're just doing one independently. Is that right?
Yes. There actually exist 3D MRIs. What we're doing is we're treating each of these. We're taking each of these slices independently because the between slices is known to be quite noisy. So in like radiology, so the radiologist that we worked with for this suggested that we really sort of like take each slice individually because the actual between slice emission can be kind of unstable.
Okay. And so those slices now, so they like a square grid,
right.
Which is a binary.
Yes.
And you're rotating that, so you have like a filtered cubical complex, I guess.
I suppose so. We didn't really think about that on that terms, but I suppose, yeah, that's right. Yeah.
And you're doing cell sets of like either the black part.
Right.
I see. Yes, where homology is changing, Euler characteristic is changing, and 72 directions.
Yeah.
For each slice now you have 7200. A vector are 72,100. I guess they're integer valued, right?
Yeah. No, because we subtract the mean.
I see. Okay.
And then
you have 25 of those.
Yeah.
For each object.
Yeah.
Okay.
Yeah.
All right. Thanks a lot.
Yeah, no problem. This is how we build the vector.
Can you hear me?
Yeah.
So for the 25 slides, each action are you using fixed AI, fix the value.
For the, we do it for the entire slice. We do it for the entire square here, basically.
Oh, God, thank you.
Yeah, no problem. So that's how we actually build the statistic. Now, the goal we had for this was to actually predict clinical outcomes. So we want to integrate this into a statistical model which predicts clinical outcomes. So the two measures of clinical outcome that we looked at are overall survival and disease-free survival. So here's a little bit of this is a sort of just an image here which describes what happens when you get cancer. So here, you're sort of, the state of your cells is pretty normal, and then there's some sort of oncogenesis here, which is your oncogenic phenomenon, which gives you cancer, basically, and then eventually your tumor progresses. Okay, when your tumor gets big enough, you're diagnosed with cancer. Okay, and then you receive radiation therapy here to treat it, and this tumor goes away. But what happens, because there's very often in cancer, the main problem with cancer that we have nowadays is the relapse. What happens is that there's some sort of, so known as tumor heterogeneity going on, which basically means that this tumor is made up of not the same oncogenomic effects, but there's actually some heterogeneity going on, which means that there are some aspects of the tumor which actually survive, which are able to cut the radiation therapy that you get, so that it can continue in your system, and then you get relapse. So this is what relapse happens here, and then afterwards you maybe get some more treatment, so chemotherapy, for example, and then eventually it just goes on to metastasis, and then most of the time the patient dies. So this is sort of like the sort of timeline of what happens from when you get cancer and what happens when you're treated, and when there are cases of relapse. So overall survival measures basically this time point when you get treatment until death, until the patient dies. So until sort of, for all practical purposes, sort of infinity into the oncogenesis here. And then disease-free survival measures the time between your successful treatment and your relapse. So it's only this interval that disease-free survival is interested in. So in sort of predicting these two clinical outcomes here. So we do this by with a linear mixed model, so a sort of statistical approach here. So first, let's get a summary of data that we have. So the data is the source is publicly available. So it's from the Cancer Genome Atlas, which is a publicly available archive of patients from all over the U.S. have 33 different types of cancer. All their information, including molecular data and their sort of clinical outcomes and sort of genomic information that you would get, molecular information that you would get from patients with cancer. And these are linked with the Cancer Imaging Archive. So the Cancer Imaging Archive is it's another public database where you have the images of all of the tumors that are matched with the patients that you get on the Cancer Genome Atlas. So all this is publicly available, and this is what we used. And we got a total of 92 GBM patients. So we're looking at the largest available set of data of patients that would have their molecular data. We looked at gene expression data as well as images as well as their clinical outcome. So we have a total of 92 patients in the end. And so we have their imaging data, which comes from the Cancer Imaging Archive. We segmented and computed these smooth Euler characteristic transforms, which means that we got our 7,200 vectors. So we have too many 7,200 vectors, so for each patient. We also got gene expression data. We applied an existing batch correction method, which basically takes all your gene expression data and it throws out the genes that don't vary. And so in the end, we ended up with 9,215 genes. Then we were also interested in sort of the whole, the whole sort of summaries of tumor shapes that exist already. So I mentioned before in radiogenomics, there are existing results in radiogenomics that basically rely on mostly geometric features. So we have morphometric data, which really corresponds to sort of shape information about the tumor, and the volumetric data, which corresponds to really the volume. So these are sort of geometric ideas here. So we took existing morphometric data, which is also publicly available from a previous study, those values that they calculated, and then we also did our own sort of volumetric calculations, which basically amounted to calculate volumes of pixels and things. So all the different sort of volumetric summaries of our data of the it's a Bayesian linear mixed model. So here we have Y is going to be our phenotype. So this is the clinical outcome, and then C would be the gene expression data. S corresponds to the topological summary. So this is our shape data. N is the morphometric, and V is the volumetric, and then we add in perturbation here. So basically, this is the Bayesian mixed model that we apply, and then we have all of our random effects, the genes corresponding to gene expression, topological summaries, morphometric summaries, and volumetric summaries, following multivariate normals. And then the variance is defined to be some sort of similarity matrix. So I'm going to give just a sort of an example here of the genetic similarity matrix for the gene expression data. So this is a quantity that's sort of known in for biostatistics. The genetic similarity matrix basically takes a sum of X, which is going to be your genotype matrix, and then scales it by the number of genes that you actually see in the end. So the sort of intuition about the genetic similarity matrix here is that it sort of models how similar patients are among each other in terms of their genotype. So genetically similar are patients to each other. Okay, so we took this idea and we sort of adapted it for all of the sort of shape features that we're looking at also. So for the topological features, we use the stacked smooth Euler characteristic transforms that we calculated for each of the patients, and also do this similar sort of similarity matrix here. And the intuition for that would be how topologically similar are patients' tumors within each other. So
Anthea?
Yeah.
The answer is the number of individuals?
Yeah. Yeah.
And so the data, like the expression data, the imaging data, and so on, those for each of the subjects?
That's right.
And those are all vectors?
Yes. What matrices, right? The G here?
Yes, I'm just trying to actually going back to the very first line here. It seems to me that like G, S, M, V, and that maybe they don't have the same dimensions, don't they? Is that?
We have one for each patient, basically.
Does the plus denote concatenation of vectors or something?
No. So we have, for our Y vector, it's going to be a vector of clinical outcomes. So there's only one clinical outcome for each patient, right? So I need two. Right. Okay. So basically, our G here, this is going to be a random effect, but we have one which will distribute for each patient, right?
Okay.
So patient has one clinical outcome, has one random effect corresponding to gene expression, one random effect corresponding to topological summaries, one random effect corresponding to morphometric features, one random effect corresponding to volumetric features, and then noise.
I see this is a real number.
Yes.
Okay.
Yes.
And now for each patient, you have a list of all of its expressions or something?
That's right. We only took gene expression in this case here. There are other molecular data available, but we only studied gene expression in this one because our interest was really studying what are the contributions of shape factors of your tumor. So varying the molecular data, we just took one kind of molecular data and held that constant, and then tried to sort of get an idea of how your prediction of clinical outcome performs based on the different kinds of shape features that you integrate.
And that's coming, the gene expression data is coming from the tumor, is that right?
Yeah, so it comes from the TCGA, right? So this happens when you in your surgery, you sequence. I mean, you basically get your tumor, and then after that you sequence it. So this you get gene expression data from.
I see.
Yeah.
Okay.
So this is our large model here. Okay, so this is our large model with all of the different sort of data types that we're looking at. And then what we did is we subset this model to try and sort of get an idea of what are the different contributions of each of these types of imaging features that we that affect the clinical outcome and our prediction of the clinical outcome as we sort of integrate them in. So we studied 12 models by subsetting this larger model here. So we started off with a gene expression only. So gene expression only is really just Y with this G vector, and then everything else deleted except for the noise here. So that's going to be our gene expression only, which is the baseline. So that is not for radiogenomic studies, but that's really just for genomic studies of cancer. So what sort of exists so far is you're only looking at trying to determine the clinical outcome of the patient based on molecular data alone, out of what Cancerology did up until we introduced these ideas of radiogenomics, which is imaging data as well. And then we look geometric and morphometric. We did this in combination with gene expression as well as that only. And then we did topological only, and then we combined it with everything else as well. So in gene expression, volumetric, morphometric, that kind of data all together. So basically what we did is we did this systematic analysis where we took this larger model and we subset it and we tried to predict performance in relation to the gene expression only, which is our baseline, according to what people do in cancer biology. So Bayesian linear mixed model, we have to specify our priors and our hyperpriors here. So we took this, which is sort of a standard approach in biostatistics when it comes to genetic data here. So it has this prior, which follows the gamma distribution, and the hyperpriors, which is the kappa 1, kappa 2 here. What is standardly done in biostatistics is to look at the leading distribution, where we have kappa 1 and kappa 2 go to 0, which basically means that we have improper priors, but it's out that in the limit, when you're looking at this distribution, the posteriors are actually proper and stable. And the reason why we do this is because we want to integrate stability effects in terms of gene expression. So gene expression here, we actually have 9,200 something genes. But since it's a large data set, and you can also have genes that actually get integrated depending on what batch correction methods you use, then want something that's stable in the end with respect to the number of genes that actually integrate in the end. So the known standard approach for biostatistics when it comes to studying gene expression, so this is the priors and the hyperpriors that we studied, that we imposed. And for posterior inference and sampling, we obtained our posterior samples of our parameter vector here using MC via a pretty standard Gibbs sampler here. And when we did the conditional prediction of our unobserved phenotypes, we did this by 80-20 splits, so and then 500 iterations. And what I want to point out here is that this here, when we do our conditional prediction here, what we have here is for each new patient, we want to be able to predict the unobserved phenotype based on their genotype here. So this is their gene expression, but also we also integrated here their the imaging information from their tumors. So there might be some sort of objection that we're sort of cheating here, but it turns out that we're going to predict the based on the gene expression data. It's reasonable to assume that we are actually going to have the imaging data, because in the case of GBM, because it seems highly unlikely that you're going to actually perform surgery on a patient and get molecular assays of the tumor without first sending them to giving them an MRI, right? So basically, first being able to get radiological information first before we actually get molecular information. So in this case, as far as medical procedure goes, it is something that's reasonable to assume, and this is something information that we can then integrate into our model in order to improve our predictions. So predictions, we measure the predictions, the performance of our predictions by studying, look at root mean square error of prediction. And what we did also is for each of these iterations that we did, we counted the number of times for which the model, the particular model out of the 12 models that we subsetted, exhibits lowest root mean squared error of prediction. So this is what our prediction results look like here. I know that this doesn't look very informative because we don't have the specification. I didn't give you the full list of all the 12 models here, but so the details can be found in our papers. But I first wanted to sort of give you an overall picture and sort of explore what the overall idea is in this. So what happens here? So all of these starred models here, these are where we integrate topological information. So the starred models here is really sort of the new contribution that we have, which is integrating the topological information of the tumor. One, this is going to be our baseline with gene expression only. And then these combinations here without the stars, these are various combinations of the geometric and volumetric information in combination, well, alone as well as in combination with the gene expression. So here is sort of stuff, this is sort of stuff and results that already exist in the field of radiogenomics. So here from our results. So this here is the tabular version of results, and here are the box plots that we see for this. So I know that this is kind of hard to see, but basically correspond to the way and the order in which we subsetted our model. What we took is the gene expression only to be our baseline. So this is going to be our 0 here. And then we looked at the relative root mean squared error prediction difference in relation to that baseline model here. So here you get an idea of how the model and how the predictive performance changes as you start to integrate more information, right? Different kinds of information. So here is it's worse if it's higher and better if it's lower, because we want small root mean squared error prediction. So basically we want small values of that. So go means better, up means worse. So we can see here is all of the stuff up until model 8 here. So model 8 is going to be this first blue bar. This is where from model 8 to the right, this is where we integrate all the topological information. And thing before here, this is just going to be existing geometric information that we see. So if you see in terms of disease-free survival, until here, we're able to replicate existing studies. So the existing radiogenomic studies, which tell you that integrating the geometric features of your tumor gives you a better better predictive performance in terms of clinical outcome. So we do this here in disease-free survival, and also see it here in overall survival here. What's interesting to see between these two here for the clinical outcome is that the topological features, which starts from this blue bar onwards here, they on average are lower than the box plots here for the geometric features only. So they're really telling you that if you integrate topological information into your analysis, then you do better in terms of predictive performance. And it turns out here, what's in bold here is our is the model 10. This corresponds to topological information only. So this one here has the lowest bar. This one has the lowest bar. And this is telling you that in terms of predicting clinical outcome, the information, so predicting clinical outcome, if you're predicting disease-free survival, the topological information is what increases your statistical power the most and has the most influence on what you do. In contrast, if you're looking at overall survival here, the topological information is basically from here onwards, right? And you see it actually does not do any better than gene expression alone. And it turns out that the geometric information here, the bars there are still lower, which basically means that the geometric information really is better than the topological information when you're predicting overall survival. Okay. So basically, we have this. This was a really interesting result that we saw here, and we started to think about it a little bit more, and we think why this might be the case. So, in terms of the two measures of clinical outcomes that we have, which are disease-free survival and overall survival, so they're both quite noisy measures, but the free survival, it's comparatively more robust prognostic measure because remember, disease-free survival means the period between when the patient is successfully treated for a tumor and when the tumor comes back. Okay, so disease-free survival in general is really when you get treated for the disease and that same disease comes back. Overall survival, it measures time from when you're treated until you die. Okay, so in terms of looking at cancer relapse and cancer recurrence, it's less noisy. So I'm not saying that it's not noisy. It's still a noisy measure, but it is less noisy because what you're really looking at is for when the tumor relapses and when you get the recurrent tumor. Where overall survival, in the end, if you've got cancer and you get treated for it and you die in the end, it's not clear what you died from. You could have died from the cancer itself, but if you get treatment, then it's known that if you have radiotherapy and chemotherapy and the sort of treatments that exist so far for cancer, they also heavily weaken your immune system. So immune system is down, then you're more prone to get sick from other things as well. So you might die from actually having a weakened immune system as well. Or you might die from the cancer metastasizing, right? So if you have metastasis, then you have a cancer which basically spreads to the rest of your body, and then you don't know exactly what kind of cancer attacking where in your body that causes you to die. So in case, there's known to be sort of 6 hallmarks of that. So it's hard to distinguish between these if you're looking at overall survival, and then finally what happens is you're looking at the end result that the patient dies. With disease-free survival, you're really looking for that particular and it's something that's important in GBM because remember, as I said before, there is no treatment for recurrent tumors. I mean, there is treatment, but it's not successful, basically. Basically, disease-free survival is influenced by shape features, which is including inherently topological phenomenon. So we're looking at the tumor coming back, right? The existence of that tumor is something that's really sort of captured better by a topological understanding rather than a geometric understanding. And if you're looking at overall survival, though, so overall survival, if you're looking at the cancer alone as the possibility that that's what causes the patient to die in the end, then geometric phenomena are really essential to include because geometric phenomena include how large the tumor is, as well as the exact location of the tumor within your brain. So if your tumor is close to one of the crucial regions for functioning, basically, and it gets very, very large, then the really, really important things are going to encompass the sort of information about the aggressiveness of your tumor. And so this here, if you're looking at the end result of your dying, right, including the geometric information is really essential. But that being said, the disease-free survival and looking at these topological features is really interesting because it also, so first of all, it sort of opens up the pathway for exploring what exactly molecular activity is going on in terms of relapse and resistance mechanisms for the actual cancer. But you can also see that there might be possible correlations between the topological features of the tumors and tumor heterogeneity. So remember when I said tumor heterogeneity is when you get relapse, because when you have tumor heterogeneity, it means that there are aspects of the tumor within the first occurrence of the tumor, which are actually different from the rest of the cells. So the rest of the cells that actually get killed off by radiotherapy, then the only way that the tumor is going to relapse is if there's some mechanism within the tumor which is actually resistant to that therapy. So this idea of integrating the topological features is sort of another measure of way that you can sort of delve deeper into looking at and studying the evolution of cancer as well as the tumor heterogeneity. So in conclusion, what we did was we counted a vector valued topological summary statistic, allowed it to be integrated into regression models, and then we applied this to GBM, and we found that this improves disease-free survival, and this sort of uncovers new directions and provides a useful measure to study cancer heterogeneity and evolution. So in terms of future work that we're looking into and that we're actually working on right now, there's lots of interesting biological questions that you can ask from this. So, for example, you can hold the shape features constant now that we know what the contribution of all the shape features are, and then vary the molecular data. So we can use DNA methylation, mutation status, or copy number variation, for example. So this is all information that's available on TCGA, which is publicly available. We're actually collaborating also with a lab in Korea that are very known for GBM, and they're providing us with information on whole exome sequencing and whole transcriptome sequencing for tumors as well. So it's very interesting. You can compare shapes of the tumors. So in GBM, also, what's known is that there's this idea of true progression. So true progression is sort of what happens when you have a tumor that actually grows because of the oncogenomic effects of GBM, but which is that the tumor looks like it's getting bigger, but we don't actually know whether it's because of the cancer effects or because there's sort of, immune systems going on which cause sort of like inflammation, and so it looks like the tumor is growing bigger, but actually it's sort of like your own sort of and the therapy and immune effects in there to try and sort of fight the cancer. And then we also want to look at gene set enrichment analysis using shape statistics. So gene set enrichment analysis is basically a sort of score to pathway, which sort of with all of the pathways, we sort of give you a numerical value for the pathway dysregulation. So what we can do then is sort of study the difference between the shape variation as well as the pathway dysregulation. And in terms of topology, given that the transforms are both injective, this implies the existence of an inversion, right? So this basically means that theoretically speaking, you can actually reconstruct the tumor from the topological summary information. So if we can do this, then what we can do then is we can do variable selection on the molecular markers that we see, so that we can do the molecular markers which are active in terms of cancer, and then what we can do is we can pick those out, try to reconstruct the shape, and see what the molecular markers have in terms of impact on the physical form of your tumor that you're going to get. I wanted to mention all the resources and references here. So except for our segmentation algorithm, our research here is fully reproducible. So we have all of the code available online for reading in the images, calculating our statistic, also running the model, and fitting the model, as well as doing the batch corrections as well. All of that is on there. The data is publicly available from TCGA and TCIA. So all of this is also, we have the segmented images on our GitHub also, if you want to try and reproduce that. I mean, you can get it from TCGA if you wanted to, but you can also just get it directly from there if you want to play with it. Yeah, what I mentioned with the segmentation algorithm, that's actually up there because we're working on some, we're trying to patent that actually, because we're trying to make it sort of user-friendly for surgeons and radiologists to be able to actually use. That's the only thing that's really not on there. So as I mentioned, the code is available on our GitHub. Details of everything is in our paper, and sort of the references and all of the existing results that I talked about here, including pictures and things, they're also referenced in the paper. I wanted to sort of mention and thank my collaborators here. So we have Lauren Crawford, Andrew Chen, Cheyenne Mukherjee, and Raul Rabadan. So these are sort of all the collaborators on this work. And I also need to thank the National Cancer Institute for giving me this grant to work on this idea. So that's it.
All right. Any questions for Anthea? So the one part I was interested in is the segmentation algorithm, because I mean that's one thing that I think we have the least kind of expertise to do the things all the things that you talked about. So I guess it's non-trivial because you're thinking of looking into patenting it. So
Right.
are there like standard tools for doing this kind of thing?
There are. So there are some methods that exist so far which are completely user specified. So it's somebody who's a radiologist or somebody who actually has experience with these things to know what they look like to be to do that manually. There's also automated ones which basically use algorithms for that. And so what our segmentation algorithm does is sort of like does both. And I mean, there's also the one thing that I mentioned also is that we only looked at one of MRI imaging that we have, which is basically T1 contrast. So there's lots of different imaging modalities that exist. So there's lots of different sort of segmentation algorithms based on what kind of imaging modality that you're looking at. But ours only really looks for T1 contrast. So there's roughly speaking, from what I understood of the different kind of imaging modalities, is that the images show up when you have grayscale. Then what tends to be darker and darker really depends on whether you're looking at fluid or something solid. So you completely different sort of backwards images depending on what modality that you're looking at. But ours actually applied to T1 contrast. So T1 contrast is where IV, the patient gets some sort of dye in his system, and so that basically goes up to the blood as well, and so that's where you can pick out the tumor better. But one thing that I should also mention also is that this with T1 contrast, if you get a good image of your tumor, that really only works because there's been a disruption between the blood-brain barrier in your brain, because that basically allows fluid in, because otherwise the dye would not be able to get into the tumor otherwise. So in this sense, this is a sort of source for confounding. So this is like a sort of a counter for our information here, because the tumors that we're looking at, the images that we get, really only where the blood barrier has been disrupted. And this is also a biological phenomenon which is known to be sort of make your GBM more aggressive. So this is sort of one of those cases where have some sort of confounder in our model, but we don't really know how to deal with that, because as far as I know, there's no batch corrections for imaging data so far. So that's sort of one thing to sort of state and put out there.
Thanks.
Yeah.
And I guess you haven't been able to look at trying to see what aspects of the topology is contributing to the or predicting the disease survival?
No, that's sort of what we're working on next. I mean, that's what we would like to do. I mean, it was very exciting to see this, because this was the first time that anybody actually really thought about sort of topological characteristics to incorporate into radiogenomics. At the same time, I'm not sure whether this is sort of this might only be relevant to GBM, right? Because as I mentioned, GBM has those characteristics where you get necrosis and multifocality, so where you actually get sort of topological features that are well defined. So if you're looking at H0 versus H2 homology, I'm not sure about what other kinds of cancers look like, whether they have that or not.
Okay. Any questions? All right, let's thank Anthea again for a great talk. Thank you very much.
Happy holidays, everybody.
Anthea, I actually had a question. I'm sorry.
Oh, hey, Patrick.
Hey, so I'm looking at the original paper, and it seems like the continuous or the similar characteristic is now a function of nu, correct? So you've translated this, rotating it around the circle and doing this integration. You now have just this is one-dimensional function in terms of nu, right?
Right, because what we did is we just sort of, we assumed that we just sort of take one tumor. We study one tumor at a time, which is sort of reasonable because we only look at one patient at a time, basically. So.
And you'll have one function now for each homology group. Is that, oh, I'm interpreting correctly?
No, because it's the Euler characteristic. Don't forget the Euler.
Oh, yeah. So it's just one function then.
Yeah. So this is why it's also important to note, right, that has the injectivity only because we're looking at enough directions. Okay, so you fix only one direction, you're not going to have injectivity because the Euler characteristic counts in an alternating sum. You're connecting components, loops, and voids, right? If you think about it, you only have one equation for 3 unknowns. So there's you're going to be able to find out exactly how many of those that you have. So yeah, so basically all homology information is incorporated in this one summary, which is the Euler characteristic.
So I guess I don't see how it would be possible to go back to specific homology information, correct? Would that be one of the challenges that you're working out now from this?
Well, yeah, I guess if in terms of sort of like looking at you know, constructing the shape. So that is hard. Yeah, that is. But I mean, because it's injective, right? This basically means that mathematically speaking, you do have an inversion. Yeah, so it exists. It's just hard to do. But you know, we're hopeful. You know, theoretically, if you can do that, then that would be really great. Because, like I said, then after that, you can look at all of these molecular markers that you have, do variable selection to try and find what are the pertinent ones, and try to see if that actually has any effect on, you know, for example, multifocality or necrosis.
Yeah, this is actually very similar to the problem I'm thinking about with my research in terms of how do we break this down and be able to isolate the parts of the topology that are actually leading.
Okay.
I think this is, I'm looking at it from a different, using functional data as well, but just looking at the homology groups individually. So
okay.
I'd be interested in talking about this more with you guys.
Yeah, definitely we should talk about this. Yeah, that would be great. Yeah.
Right.
Okay, thank you. All right, again, everybody, and you know, happy holidays.
Thanks.